Hey, so today we are going to talk about viruses, specifically foundational chemistry in viruses and viral respiratory illness treatment. So let's first define what a virus is. A virus is a small, simple creature which can or can't be classified as a living organism. There are much debate whether a virus can be called living. Most viruses contain three components: nucleic acids, protein, and lipid coating. Nucleic acids, either DNA or RNA, provide genetic instructions for future viral reproductions. The reason that a virus can be contagious or spread throughout your entire body is because it reproduces so quickly. And it has a protein coating to protect the nucleic acid and a lipid coating surrounding the protein coating, but it isn't present in all cells. If they do have a lipid coating, it's called an envelope virus rather than a naked virus. This is a picture of a T4 bacteriophage. It has the red lines is the DNA, you have the protein coat, you have the sheath, you have tail fibers, and you have a base plate. This is without the lipid coating. If you see a lipid envelope virus, you have viral proteins included in the envelope, lipid envelope, protein coating, and either the viral DNA or RNA. So how does a virus function? They take over the function of a cell, or what you can call a host cell. Basically, they hijack your body, and they cannot live without attaching itself to another living thing, such as a host cell in your body. So different strains of a virus can infect different types of cells in your body and can infect different plants, animals, or even bacteria cells. So let's look at flu viruses. Whenever we think of a virus, we tend to think of the flu because we are vaccinated every year to protect us from the flu. But the flu is a contagious respiratory illness caused by influenza viruses. The word flu comes from the word influenza. And one type of flu that we are extremely aware of is coronavirus or COVID-19. Coronavirus is a type of respiratory illness. Basically, glycoproteins of the virus attach to the host cell. So the word corona comes from the word crown. So when glycoproteins of the virus attach to the host cell, it makes a shape of a crown, therefore coronavirus. Now, coronavirus, as we see today amidst of a global pandemic, it is affecting thousands and thousands of people all over the world, asymptomatic or symptomatic, and it causes a lot of problems for people that have respiratory disease, or it affects people's gastrointestinal tract. Let's go to how to prevent us from getting the flu. For that, there are vaccines. Vaccines work by stimulating the body to prepare against specific antibodies to possibly give immunity against the virus. So basically, scientists try and take a strain of a virus and try to come up with antibodies that they inject into you, or there are other ways to get a virus or a vaccine into you, such as inhalation or eating even. And basically what it does is it tries to prepare your body for antibodies. So if you do have a virus, it is quicker to fight off that virus. So a vaccine contains microorganisms that activate the body's immune system. The white blood cells, such as B cells or T cells or antigen-presenting cells or APC, recognize, destroy, and remember the pathogen. So if you do encounter the virus later in your life, your body can fight it off a lot easier because your body produces memory B cells to fight off the virus a lot quicker. So if you look at the 5 main type of vaccines, you have live attenuated virus, inactive virus, taxoid virus, subunit vaccine, and conjugate vaccine. So live attenuated vaccine fight viruses and contain a weakened version of the living virus. So the vaccine itself has the living virus, but it's just a weaker form of it. A inactive vaccine fights the virus and contains the killed virus. So because viruses can reproduce and replicate so quickly, inactive vaccines, they keep contained. Taxoid vaccines prevent diseases caused by bacteria and produce toxins in the body and contain weakened toxins. For example, you get a tetanus shot or a tetanus vaccine. People usually get it when they're infants. Subunit vaccine include only essential antigens of the virus. So it doesn't include every part, only a few strains. And then conjugate virus, they fight bacteria that have antigens with an outer coating of polysaccharides, which this hides from the child's immune system. So what this vaccine does, it connects a polysaccharide to the antigen. And the antigen is obviously the antigen is what the antibodies fight. So that causes an immunoreaction. So let's look at the type of flu viruses. You have hemagglutinin, a glycoprotein that enables a viral particle to dock before it enters a host cell. So basically tries to block the virus before it comes in contact with the host cell, or a neurodiamine, which is an enzyme that catalyzes a cleavage reaction to allow new viral particles to escape from the host cell. The enzyme snips off the sugar molecule, or sialic acid, from the glycoprotein on the surface of the host cell membrane. If you may not understand this, take a look at this diagram. So you see so let's start from right here. So the yellow is the sialic acid, which is on the edge of the cell. You have the red, which is the hemagglutinin, and the neuraminidase. So the virus is entering the cell, which basically is acting as a substrate, attaching itself to the active site because they are enzymes. And then it comes into the body, and then the RNA replicates in the nucleus. Then it comes back, so you see D if you're following that. Right here in this area right here, the virus is replicating. So it moves along this way, and it's beginning to detach from the host cell. But then if you see here, you see the neuraminidase inhibitor, which is a black dot, which inhibits the neuraminidase enzyme. The reason why you don't see a hemagglutinin inhibitor is because neuraminidase seems to be a better target for drug design and has become a focus for research. So the chemicals that interfere with this binding are the inhibitors that have a specific fit with the enzyme. So we looked at what a virus is, we looked at the function of a virus, we looked at the types of vaccines, the types of flus, how a virus interacts with the body. But let's look at how a body actually fights off the virus. So if you look at this, if you remember the sialic acid, which was on the border of this the white part, you'll see that is what is it basically it is trying to prevent the virus from infecting the host cell or reproducing. So if you really look at celiac acid, you can see it has it's just chemistry. So if you look at the pink, you see a hydroxyl group. If you see the darker purple, you see an ether. If you see the lighter purple, you see carboxyamide. So when neuraminidase inhibitors were first designed, the Relenza vaccine was first made. They have a chemical structure similar to sialic acid, so they are able to bind to the active site of neuraminidase. The class of drug is active against both influenza A and B viruses, meaning it is active against hemagglutinin and neuraminidase, and is claimed to reduce the symptoms of flu and shorten the time of its effects, but must be taken within 48 hours of the appearance of the symptom. So now, if we look at the Relenza, we see it has the hydroxyl groups, it has the ether groups, it has a carboxyamide group. The only difference is it has a carboxylic acid right here. Carboxylic acid right there. But because this vaccine is so similar to sialic acid, it's able to fight off the virus. So now we have seen the chemical foundational chemistry of a virus. We have also seen how chemistry is related in the treatment. So because scientists were able to produce an inhibitor that was so close to sialic acid using chemistry, they were able to fight the virus called the Relenza flu. And then we also looked at biochemistry, how a substrate, an active site, and an inhibitor. So the three main chemistry subtopics that I used was medicinal chemistry, antiviral medications. I used enzymes from the biochemistry chapter, and I used organic chemistry, which is shown right here. That's it. That's foundational chemistry in viruses and the treatment of viral illnesses. Thank you.